Hi there, and welcome to this IB revision video for IB biology. And remember, this revision video is support only, so it is not a complete revision guide. Today we are going to be looking at cell division, and part one of this video is on mitosis. So first thing we're going to look at is something called cell theory. So cell theory is the idea that all new cells arise from pre-existing cells to form by mitosis. And for example, you started off as a single cell, the zygote, when the egg was fertilized, and then mitosis has happened over and over again in order to produce you now being a multicellular organism. Now the key thing here is mitosis is actually just the events that happen in the nucleus, and it's part of something bigger called the cell cycle. So mitosis is a type of what we call nuclear division. All cells except gametes contain an identical set of 46 chromosomes in humans, and they all undergo mitosis to grow. Obviously we'll look at meiosis afterwards. The purpose of meiosis is to allow growth, replacement of cells, and asexual reproduction. So first of all, we need to understand that cell proliferation is an increase in cell numbers. So this happens in animals and plants for growth. In animals, proliferation, a rapid increase in numbers happens in 2 points in your life: embryonic stages and the juvenile stages, and that'll be just parts of your body. So you suddenly get the bones grow, for example, and you get an increase in bone plates at the end of your bones. That's when you get a growth spurt. In plants, only cells in the tips of shoots and roots called meristems are able to do mitosis, and cells in meristems divide, but ones outside meristems will just elongate to grow. We also use mitosis in cell replacement. As you'll be aware, skin cells are being replaced constantly, and they're actually made in the basal layer of the epidermis, so down here, and they are constantly displaced towards the cell surface by continued division, so cells that are produced after and push them outwards. During the transit, they produce a large amount of tough protein of keratin, which is hydrophobic and prevents water getting into the body. Cell replacement is questionable as cell proliferation, as cell production is not faster than death. So what we're saying here is it is not necessarily that this cell replacement isn't actually proliferation, it is more likely to be replacement as a result. Okay, so production and death are at the same, are at different sites in the. Another use of proliferation is tissue repair. Damaged wounds need new cells to heal, so we use a cell division to heal. This involves undifferentiated stem cells, and they can divide and then differentiate. Number of stem cells in tissues mean that some are able to repair themselves more than others. So this is what happens originally when we have a cut. We block it off with platelets, and then new cells grow to replace the damaged cells. The skin epidermis is therefore a good example of tissues that are able to repair itself. So before we do about mitosis, we need to understand what a chromosome is. So before replication, each one of these are different chromosomes. So if we use a bit of the sexist way, I suppose A will be the male chromosome, so the one you get from your dad, the paternal one, and that one is the female one. They are both two chromosomes, so they are what we call homologous chromosomes, one from your mom, one from your dad. A chromosome is DNA that is wrapped around a histone protein, and after interphase, when DNA replicates, we end up with two sister chromatids which are identical. So this chromatid here is identical to that chromatid. This chromatid here is identical to that chromatid. We still say there are only two chromosomes there, but they are double copies of the chromosomes. And this all happens within the cell cycle. So here's two different versions of the cell cycle for you to see. But if we use this one here, the cell cycle is mainly the process of interphase. So interphase is when the cell grows, okay, when the DNA is replicated, and then what happens after the growth, where organelles are replicated, so you've got the organelles ready. Only a small percentage of the cell cycle is mitosis, and mitosis is made up of 4 stages, which we'll look at in detail shortly. So majority of the cell's life is outside of mitosis, only a small amount is in mitosis. So the first stage, as we said, is interphase, which technically isn't mitosis, and then we have 4 more stages in that. So interphase, prophase, metaphase, anaphase, and telophase. And you need to be able to describe the events at each of these stages. So let's have a look at each of them. So technically interphase is not part of mitosis, it happens before mitosis starts. But during interphase, what happens is a signal reaches a cell and it makes the DNA replicate from single strands to double-stranded chromatids, sister chromatids, identical sister chromatids. The cell makes copies of its chromosomes and the cell will otherwise just continue normal activity. Remember, there are different stages of interphase. So there is what we call G1 stage, and that is where the cell grows and it makes proteins in order to do DNA replication. You have S stage, which is where the DNA replicates, and you have G2, where organelles such as mitochondria, ribosomes, etc. replicate ready to become 2 cells. We used to call this the resting phase, and the reason we used to call this the resting phase was because you can't see any of this replication of DNA happening. You can because the DNA has not condensed at this point. So, as I said before, interphase is several stages. It used to be called the resting phase because you can't see it, and it's and in stage 1 the cell grows bigger, makes proteins ready for replication of DNA. In the S phase, the proteins are replicated, and in the G2 phase, the organelles are replicated. We then go to prophase, and in prophase, that is where the chromosomes start to become visible. So you can see here, prophase, the chromosomes are starting to become visible, and they'll become thicker and shorter, and you can now see them under an optical microscope with a stain. And an example of a stain that you might use is methylene blue to stain them. Nuclear envelope will also break down, so this bit here, this envelope will break down. Now we're in mitosis, we're just talking about events in the nucleus, but the nuclear envelopes are going to break down, and the centrioles, which are proteins, move to opposite poles where they start forming spindle fibers. In metaphase, those spindle fibers attach to the middle of the chromosome, which we call the centromere. So that is this area here. Okay, here and here, and the chromatids attached to them, pulling on the spindles attached to the centromere, pulling on the chromosomes, making them line up along the equator. And here you can see a typical picture of that using a micrograph and electron microscope. I'm sorry, a light microscope, not an electron microscope. Anaphase, then, the sister chromatids are pulled apart and they move to opposite poles of the cell. So the spindles contract, pulling the chromatids apart, and the sister chromatids, which don't forget are identical copies of each other, go to the opposite poles of the cell. So they become separated. At this point we don't have 2 cells, but we have chromosomes now moving to opposite poles. Once they are separated, the sister chromatids become individual chromosomes. And you can see here how that would look on a light micrograph as well. And the final stage is telophase, which is when the chromosomes reach the poles, and then the first thing that happens is the nuclear envelopes reform. So you end up with 1 cell at this point with 2 nuclei in it, and the chromosomes then we stop being able to see them because they become long and thin, so they are no longer visible in the cell. After that happens, and this is often linked in questions, cytokinesis is followed, and this is where 1 cell becomes 2. So you might start off with 1 cell with 2 nuclei, and then what happens is the membrane begins to fold inwards, and eventually the membrane in an animal cell separates the cytoplasm to become 2 cells. In a plant cell, you have your 2 nucleuses, and what happens is vesicles start to form along the middle, and eventually they form a new cell membrane, and then a new wall is laid down. So this is a key difference between animal and plant cells in mitosis. Another difference, which I forgot to mention earlier, is that in animal cells we have centrioles that move to the poles and make spindle fibers. Plant cells do not have centrioles and have a different mechanism which you don't need to know about to make their spindle fibers. So the second part of cell division then is called meiosis that we want to look at, and this is the production of gametes. So gametes, as you should be aware, are egg and sperm cells or pollen in plants. So again, we need to be familiar with these terms. So this is called a karyotype, which shows you a map of the 23 pairs or 46 chromosomes in humans. And you should be aware there are autosomal chromosomes, which are the first 22 pairs, and then the 23rd pair decides whether you're male or female, which is the sex chromosomes. And a homologous pair of chromosomes means that you get one from your mom, one from your dad. So this pair 22, one of those is the paternal chromosome from the dad, one of those is the maternal chromosome from the mom. And obviously when we're making gametes, we don't give all 46, we only pass on 23 of our chromosomes. So when you have a full 46 chromosomes, this is called a diploid cell. And when you have 23, because you've only got one of each homologous pair being passed on, so for example, we could pass this one on or this one, it is called a haploid cell or haploid chromosome number. So diploid possess a full set of homologous chromosomes, haploid only pair one set of chromosomes. So you should be able to look at this and think about this question. So if a gorilla even has a diploid number of 48, the brain cells will have 48 chromosomes, the epithelial cells will have 48 chromosomes, the muscle cells will have 48 chromosomes as they are diploid. The haploid sperm will have 24 chromosomes. So we need to understand what meiosis is. So meiosis is, as we focused on with mitosis, is the event in the nucleus, and meiosis is reductive division in the nucleus. It results in 4 genetically different daughter cells, okay, and they are haploid, which means they only have 23 chromosomes in for humans, 24 for the gorilla, and that means they're making gametes for the egg and sperm cells for reproduction. So we need to know the stages that happen in meiosis. And meiosis can be split into 2 stages, the first mitotic stage and the second stage. So if we look at the first stage, we have prophase 1 to metaphase 1, anaphase 1, and telophase / cytokinesis 1. So don't forget, interphase has already happened, as in the cell cycle that we just talked about. So interphase is when the DNA has replicated. So you can see here the DNA has replicated into sister chromatids. In prophase 1, each chromosome has already duplicated, I should say, and it remains closely associated. They're called sister chromatids. The homologous chromosomes pair up. So here you see that could be pair 1, that could be pair 2, that could be pair 3, for example, of the 23 pairs. And they have to pair up to make sure you have the right full set of genes in each cell created. Metaphase 1. So metaphase 1, we have the chromosomes lining up along the equator. So again, the homologous chromosomes line up along the equator. Now notice when the chromosomes pair up, and it's not very clear here, but we'll talk about it a bit later, parts of the chromosomes can overlap and swap some genetic material around. So the gene for eye colour could be swapped with another gene for eye colour, for example, in prophase 1, and that is called crossing over. So in metaphase 1, as I said, the homologous chromosomes line up along the plate. The spindles attach to them, like in mitosis, and they pull the homologous chromosomes apart. So in anaphase 1, you can see the homologous chromosomes are separated, but the sister chromosomes remain together. So the maternal chromosome, let's say, of chromosome 1 goes that way, the paternal chromosome 1 goes that way, but that is still made up of 2 sister chromatids that are identical, apart from where crossing over has happened. Now this shows another source of variation that happens in metaphase or anaphase 1, because there is independent assortment. So this red maternal chromosome could have been at the top, and 50% chance it could have been at the bottom. So for each one they can be arranged independently, and therefore you'll have different combinations. So here we've got a red one at the top, red one at the top, dark blue one, but that could have easily been blue, 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 or blue, red, blue. So we have something called independent assortment. The chromosomes are then pulled to the opposite poles, and then we have the cell splitting into two in cytokinesis. So two daughter cells are formed, each daughter cell containing only one chromosome of homologous pair. The chromatids are still attached in the centromeres. We get to the reductive stage in number 2, where prophase 2 occurs. The DNA obviously has not replicated again here at this point. The chromosomes then line up at the equatorial plate, and at this point it's the same as mitosis. So the spindles attach to the centromeres of the chromosomes and pull the sister chromatids apart in anaphase 2. Now, when they line up at the equator in metaphase, you can see if they've had crossing over, such as this one here. Again, independent assortment can happen, because if this chromosome was the other way around, the little blue bit here would be on the other side, and therefore we can still exchange genes randomly into each cell, into different alleles, I should say, into each cell. Telophase 2 is when cell division is then complete and the nucleus is reformed, and we end up with 4 haploid daughter cells. So you need to be able to state where that variation comes in. If you're not sure, you need to speak to your teacher about this in more detail. So as I said earlier on, we have independent assortment in anaphase 1. So when the chromosomes have aligned, the chromosomes are then pulled apart, and depending on which way around they were, depends on which direction they go. Crossing over happens in prophase, and that again means alleles are exchanged. The same genes in all chromosomes, but different alleles are exchanged. And after crossing over and independent assortment happens, random mating also occurs, which obviously gives different chromosome pairs to come together, and random fertilization occurs as well. Okay, now don't forget that in metaphase 1 we have independent assortment, but also in metaphase 2 as well. In an earlier video we talked about mutations and non-disjunction, making an extra chromosome appear in cells, and an example of this would be Down syndrome, where you have the extra 21st chromosome. So here you can see how this would happen. So in metaphase 2, this would be the chromosomes line up, and then instead of being pulled apart, so your sister chromatids go to either cell, one of them doesn't pull apart, and you have this extra chromosome in a gamete, which then leads to an extra chromosome in your cells. A lot of the time this will prove fatal, but Down syndrome is an example where it does not. And the final part of this video then, for higher level students only, is to just look a bit more in depth at the cell cycle and maybe the control of the cell cycle and cancer. So as we said before, the cell cycle is mainly interphase. Not all cells can divide continuously, and when we're looking at interphase, we have G1, which cell grows bigger and makes proteins to help replication of DNA. S phase, where replication of DNA occurs, and G2 phase, where the organelles are replicated, before mitosis, which is a type of nuclear division. The cell cycle is mainly interphase, very little mitosis. And as said before, mitosis is the process that divides the nucleus, after which cytokinesis divides the whole cell at a time, and it's only a small amount. There are 3 main stages, again just a bit more detail for higher level students. So G1 is after the previous mitosis, it's the growth phase before the DNA replication. The DNA is replicated in S stage, making identical DNA molecules held together in something called cohesive loops until metaphase ends. And G2 phase is when growth resumes and it prepares the cell for mitosis and the organelles replicate. So what happens in interphase? So absolutely key that you understand some of the events that happen in interphase. Cells are not actively dividing in interphase, they're carrying out normal activity. But beyond that, the following things are happening. Genes are not used in cell activity, will remain condensed in interphase. The cytoplasm volume increases. New enzymes that are going to be used synthesized. The membrane area increases by adding new phospholipids and cholesterol. The mitochondria numbers increase, and the chloroplast numbers increase in plant cells. More Golgi apparatus are made, and cellular reactions continue as normal. So hopefully, if you think about it logically, these events have to happen because you need more mitochondria, more chloroplast, more Golgi if you're going to go from one cell to two cells. There are checkpoints in the cell cycle, and these will hold the cells and prevent the cells from moving to the next stage to ensure that cells stop dividing when there is enough cell proliferation. So when we get to the point where we don't need any more cells, they're held in place by checkpoints. And there are different groups of proteins which can coordinate the sequence during the cell cycle. So there's something called cyclins, and they activate cyclin-dependent kinase enzymes, which binds phosphates to other proteins, activating them. The type of kinase activates a particular group of protein during the phase in the cell cycle. So when the cyclin binds and activates CDK, for example, CDK then activates proteins to regulate the cell cycle, and then the cyclin is destroyed. So let's have a look at some of the. So here you can see the different cyclins that control the different levels. So you can see here we have cyclin D, which activates therefore the move from G0 to G1. So you see the number of levels increase during G1. We then have cyclin E, which prepares the cell for DNA replication in G1 phase, which would be here. We then have cyclin A, which activates DNA replication in the nucleus, and then cyclin B promotes the assembly of mitotic spindles in preparation for mitosis. So you need to know which cyclin prepares which stage in the cell cycle to happen and then triggers that next stage. So there you go again, cyclin D triggers the cells to go from G0 to G1 and G1 to S. Cyclin E prepares the cell for DNA replication. Cyclin A activates the replication inside the nucleus, and cyclin B promotes the assembly of mitotic spindles to prepare the cytoplasm for mitosis. Now obviously if mutations happen, this can cause a problem. So mutations are random, they can occur at any time. Their chance is low in DNA, so it's stable and it is resistant, but the risk can increase due to mutagens that can cause errors in pairing of the complementary bases. So there are two main types of mutagens: radiation, which gives enough energy to change the chemicals in the DNA, and chemical substances that change the DNA, such as tobacco smoke, which contains nitrosamines. So the consequences of mutations are obviously quite severe. Mutations can cause a loss in cell cycle control, which can be inherited by daughter cells, and this leads to cell division becoming uncontrolled and much greater than apoptosis. So remember, apoptosis means programmed cellular death. So what we're saying here is the new cells are being created at a greater rate than the old cells are being killed off, and this develops into a tumour. Now there are two groups of genes that can change a normal cell into a tumour cell. The first type is proto-oncogenes, and they have a variety of roles in the cell, from controlling gene expression for genes to link to proliferation. Others control messenger systems, and others are receptors for growth factors. Proto-oncogenes can mutate to form oncogenes, which then actively promote cell proliferation and are dominant. So we start off with a proto-oncogene, which is the healthy version, and then a mutated version leads to increased cell proliferation. This means you only need one to start uncontrolled division, and that can just keep growing into new cells exponentially. And normally it's from a missense mutation, so where the wrong amino acid is being. So consequences of mutations then. There is the first one we said, proto-oncogenes. Second one is tumor suppressant genes, and basically tumor suppressor genes apply the brakes at checkpoints. So if you go from S phase to G2 phase and there's a problem, they could stop the cycle there. They repair the DNA, correcting errors and reducing the mutation rate. And they have a role in apoptosis for DNA that is damaged and can't be repaired. And what happens here again is you get a base substitution, so one of the DNA bases is changed, and that can lead to a truncated protein, which is one that is no longer working. It's normally recessive as only one pair of the genes mutates, but over a lifetime multiple mutations can lead to tumours, and sometimes over 10 mutations are needed. So normally what happens is cells will divide several times and then they have apoptosis, they're programmed to die. A cancer, when it becomes mutated because of those two genes that we've just mentioned, can lead to uncontrolled mitosis and then as a result. Now you need to know that tumours are not cancer, so tumours are benign, they're not cancerous, and if they're contained in one place and normally surrounded by a membrane preventing them spreading. And as a result, they are tumours. They can still be dangerous, can still apply pressure to your organs and stop organs working properly, so may still need to be removed, but not always. They are medically therefore less serious. When the tumour has spread, this is called malignant, and malignant is a cancer. So it can spread to cause secondary tumours around the body, called the process called metastasis, and all tumours would have to be destroyed before it continues to spread as a result. You need to be familiar with the early symptoms of cancer, but just be aware there are over 200 types of cancer in different forms, and people should visit their doctor if they suspect they've got cancer, because early diagnosis massively increases survival chances and will also reduce the treatment needed. The main signs for cancer are a lump, an unexplained lump, which could be a tumour, unexplained bleeding. So, for example, if you cough up blood, that could be a sign of lung cancer. Long-term cough, which isn't being fixed, again could be a sign of lung cancer, and a loss of weight without dieting. So unexplained weight loss, again, could mean that the nutrients that you're taking in are being stolen by the cancer cells.